I'm really excited to introduce Dr. Phred Benham. He's a postdoctoral researcher at the Museum of Vertebrate Zoology at University of California, Berkeley. Inspired by a childhood fascination with birds, he has pursued a career in ornithology. He received his Bachelor's of Science from Louisiana State University, Master's of Science from the University of New Mexico, and PhD from the University of Montana. He has studied birds around the world and a wide variety of bird groups, including Canada warblers and Savannah sparrows in North America, tailor birds in Southeast Asia, which, if you're not familiar with tailor birds, are called tailor birds because they actually do sew like tailors. And in the Peruvian Andes, he has studied a group of hummingbirds called the metal tails, which I think is a great name. He has broad interests in how birds have evolved and diversified into all the amazing species we share the world with today. In particular, he tries to understand the mechanisms that allow birds to persist in harsh, physiologically challenging environments like the salt marshes of San Francisco Bay.
Thank you very much, Katie and Serena. I'm really excited to talk to Birdie Hour today about one of my favorite habitats and species that live in that, and those are the sparrows that you find in salt marshes. So in general, I'm really fascinated by all the places birds have managed to colonize and adapt to, and really interested in how they evolve different adaptations to persist in some of the coldest, hottest, driest, and highest environments on the planet. And this motivation is really what has led me to study birds in a really extreme environment, which are the salt marshes, which we are pretty familiar with here in the Bay Area. And salt marshes are really dynamic habitats that form the boundary between the marine and the terrestrial realms, and they're generally characterized by they're generally found in temperate regions around the world where rivers are emptying into the sea, and where there's some protective elements like these sand dunes here that protect the marsh from wave action that would destroy them. So this allows sediments to accumulate in the estuary and provides a substrate for the marsh plants to actually take hold and form these marsh environments. And so above all else, what really makes these environments as extreme is that their life in the salt marsh is governed by the daily ebb and flow of the tides. So if you're a marine adapted organism, you have to deal with daily exposure at low tide where you're left high and dry. Whereas if you're a group of organisms coming from the terrestrial side, you have to deal with the opposite at high tide when your environment is flooded with salt water. So this ebb and flow of the tides forms this zonation you see in the salt marshes, where you have plants and animals at one end. They're much more adapted to salt water and regular submersion by the tides. So in there, you'll tend to find things like California cordgrass and things like eelgrass, which are almost never exposed at low tide. Whereas things in the higher marshes are less salt tolerant and are only submerged underwater at the highest tides of the year, usually in the spring here or late spring here in the Bay Area. And these are things like my beloved pickleweed, which you find in large mats in salt marshes here in the San Francisco Bay, and other things like alkali heath. While the tides are one of the main things that shape the ecology of these ecosystems, another important factor are the freshwater inputs into the tidal marsh. So for example, here in the San Francisco Bay Area, over 40% of California's land area is actually the water from over 40% of California's land area is draining through the San Francisco Bay estuary, primarily from the Sacramento and San Joaquin rivers, which form the Delta region here, just east of us. And what this input of fresh water, especially with the spring runoff from snowmelt in the Sierras and other mountain ranges, creates is a salinity gradient from places, here's Seasum Bay near Fairfield, where it tends to be lower salinity or brackish waters, as shown by these bluer colors. The San Pablo Bay, which is slightly more salty, to the South Bay and the Golden Gate, which is approaching seawater in its actual level of salinity, given the reduced inputs from freshwater systems and greater influence of the tides on the salinity gradient. And so this salinity gradient you find in estuaries around the world, especially here in the Bay Area, creates some really unique physiological challenges for the organisms found in these tidal marshes, both for the marine animals and for the terrestrial animals that intersect in this environment. So the central challenge physiologically when you're dealing with salinity is that almost all vertebrates, fish, mammals, birds, reptiles, we all keep the salt levels in our blood at this constant 300 milliosmoles per kilogram. You don't really need to worry about what those are. The important thing to remember is that freshwater is about 0 milliosmoles per kilogram, and seawater is about 3 times what we need to keep the salt water balance in our blood at 1,000 milliosmoles per kilogram. And this is really important for maintaining this constant level is really important for cellular function in pretty important ways. So ideally we maintain this 300 milliosmoles per kilogram, which is equal to the osmolality or salinity within the cells. And this is what we call an isotonic solution. So equal amounts of water and salts are transferred and everything's in equilibrium. If our blood gets too salty, the osmolality of our blood increases. And what this leads to is the water from our cells exiting the cell, and you get shriveled blood cells like this. This, as you might imagine, has very detrimental effects on the animal. And this is what happens when we get dehydrated. On the other side, an animal that's losing salt ions is placed in what we call a hypotonic solution, where the salt levels are greater within the cell than they are outside the cell. So what this leads to is water pouring into the cell, and the cells expand and then can rupture, which, as you can imagine, also has some pretty negative physiological consequences. And so to illustrate this further, we can think about what it would be like to be immersed in either a saltwater environment or a freshwater environment. And here, all these blue and red dots signify different molecules. So all the blue ones are water molecules, and the red ones are sodium chloride molecules, or salt. So in the freshwater, you have much more water than salt, whereas it's the opposite in a saltwater environment. So one thing that's constantly immersed in water is a fish. And if you dump a fish in saltwater, the concentration of water is much greater than the environment, and the concentration of salt is much lower than the environment. So if this fish has no way of balancing or buffering itself against the environment, what you have is a water leaving the body of the fish and salt ions entering the body of the fish. And this can lead to the fish essentially becoming dehydrated. And if nothing happens, it would probably die pretty quickly. In contrast, if the fish were to swim upstream into a freshwater environment, the conditions have flipped. So now you have greater levels of water in the external environment than inside the fish, and lower levels of sodium chloride. And then you have opposite transport. So water comes into the body of the fish, and sodium chloride leaves the fish. And so for most fish species, they're either adapted to a freshwater environment, so they are coping with this osmotic situation where they can deal with water, they can retain ions and prevent excess water from entering their body. Whereas fish in the saltwater environment have the opposite challenge that they've evolved mechanisms to cope with. And there are only a few specialized species like these coho salmon and other anadromous fish species can actually transition between saltwater and freshwater environments. And they do this by modifying all the molecular mechanisms in their gills to pump ions out if they're in the saltwater environment, or pump them in if they're in a freshwater environment. And they can do this when they make their migrations upriver. So what about birds? Birds obviously are not immersed in saltwater like fish are, but they're still often exposed to the high salinity content in their salt marsh environments through their diet, and they often drink the salt water as well. But we're lucky to live in a place like the Bay Area where we have this amazing diversity of water birds, especially during the winter. So it's pretty typical for places like this is in Alviso, where you see all sorts of shorebirds, ducks, herons, egrets, pelicans, and a pretty amazing diversity of water birds. And so these water birds all have a unique adaptation to cope with this high salinity environment. And so, for example, things like the endangered ridgeways rail, which we have here in the Bay Area, all water birds have these specialized organs right at the base of their bills called salt glands. And these are organs that evolved to cope with a high salinity environment. And so without getting too complicated, what they basically do is the individual cells within the salt glands have evolved this molecular machinery to transport sodium and chloride or salt ions from the blood into these collecting ducts that allow the birds to concentrate the salt into a very salts into a very concentrated solution, and then those salts can exit the body. And this allows the birds to maintain saltwater balance, or that magic 300 milliosmoles per kilogram in a high salinity environment. Well, this is shared among most water bird lineages, everything from albatross to rails to ducks and herons. Not all birds possess these salt glands. And chief among them are the Passerculus, which actually comprise most of the world's bird diversity. This is what we more commonly call the songbirds. And they lack these salt glands. And in many ways, they're more tied to the terrestrial environment than what we consider water birds. And so despite comprising over half the world's bird diversity, the individuals shown on this slide are most of the species that have adapted to these tough salt marsh environments. And I realize this is not a bird, but the salt marsh harvest mouse, which is another endangered species in the Bay Area, also lacks these salt glands. So what is it about these birds that have managed, these few choice species, to actually to adapt to these environments? And really, there's a lot of different behavioral, life history, and physiological mechanisms that allow them to persist. So one way to deal with high salinity is first to change your drinking behavior. So here's early experiments in drinking behavior in Savannah sparrows showed that freshwater-adapted subspecies of the Savannah sparrow, when given drinking water of increasing salinity, actually increase the rate at which they drink water. And this is a pretty common response for freshwater adapted organisms. Whenever we say, including humans, so whenever we eat lots of salty food, like chips or something, this triggers a thirst response in which we drink water to help balance out the high salt load we just ingested. However, if your only available drinking water is salt, this means you're exacerbating those osmoregulatory challenges as you're consuming more and more and more salts and causing more and more physiological problems for yourself. On the other hand, salt marsh adapted subspecies of the Savannah sparrow have been shown to respond to this increasing salinity in their drinking water, and they'll actually decrease their drinking rate. So they're consuming less salt as the water gets saltier. It's also been shown in a lot of these different salt marsh songbirds that they have increased salinity tolerance, or the ability to survive while drinking pure seawater in some cases. And this reflects the increased osmoregulatory capacity, or the ability to maintain their salts and waters in their bloodstream at constant levels. And the way a huge body of work has shown that they do this is with adaptations centered on the avian kidney. So the avian kidney looks quite a bit different than the typical kidney bean shape of the mammalian kidney. It actually kind of reminds me of a broccoli floret. And the important thing but they do function in a really similar way. So you have this outer area called the cortex, and then this inner layer called the medulla. And throughout these bird kidney and the mammal kidney, you have these things called nephrons, and then a long loop of Henle in some of these nephrons. And so what this loop of Henle allows the birds to do is to concentrate salts in the urine and excrete those salts from their body and maintain saltwater balance. It's exactly what mammals do as well. We know that these salt marsh birds are able to increase their salt excretion abilities. They increase water retention, which is also a function of the kidney. And as I mentioned, they reduce salt intake. And this is largely based on the kidney morphology. So they have overall bigger kidneys. they have overall increase in the volume of the medulla, which allows them to make concentrate salts in the urine even better than a bird with smaller medullary volume. And they have overall increased number of nephrons, so they could just doing this more and more throughout the kidney. And another mechanism that allows them to maintain saltwater balance is they reduce evaporative water loss. This is basically the water we lose through sweating, or birds mostly pant, which allows them to cool down, but it means they're losing a lot of water when it gets hot out. So that's another mechanism for reducing water loss, which I will talk about more as we move along in the talk. So these sparrows also have a lot of unique life history adaptations relative to their upland relatives. They build nests higher in the vegetation, and this is a way to avoid those high tides, so the nest doesn't get submerged in water. And when nests do get submerged in water, nestlings, as soon as like 8 days, are able to climb up in the vegetation and escape the highest tides. They tend to have smaller clutch sizes. Even the song sparrows in the bay have, on average, like 3 eggs, whereas birds in the hills, like in Tilden Park, have about 4 eggs per season. They also shift their breeding season to avoid the highest tides of the year, which in the Bay Area tend to come in late May and June. So here there's an upland subspecies of song sparrow in the Bay Area, and then a tidal marsh one. And so the tidal marsh ones have actually shifted the peak of their breeding season 2 weeks earlier to avoid the flooding from the high spring tides. And if all else fails, at least in one salt marsh sparrow species called the salt marsh sparrow, eggs have been documented surviving submersion in tidal water for over 90 minutes, which I think is pretty incredible. So finally, tidal marsh birds also are known to have much larger bills. So here's the large-billed Savannah Sparrow species, which is a tidal marsh subspecies from the Colorado River Delta in Mexico, versus a sort of typical Savannah Sparrow. And you can see that the bill size is almost at least twice the size in the salt marsh adapted population. Another really common adaptation you see across different sparrow species is that they tend to get this darker plumage in salt marshes. And this is thought to be related to matching of the darker muds that they encounter in salt marshes, and so make them less susceptible to predation. And so that's really a brief overview of some of the most common adaptations these birds have to the high salinities and flooding that they encounter in these tidal marshes. And this was really what I focused on for much of my dissertation. But as I was traveling between all these amazing salt marsh places, I got caught in a very familiar phenomenon to those of us who live in the Bay Area, and that is this just ridiculous amount of traffic and congestion. And so it became really impossible for me to ignore what the impact of 8 million people are when they're trying to share the same environment with these really unique tidal marsh sparrows. And this isn't just an impression I got while sitting in traffic, as frustrating as that might be. But over the past century, here's the South San Francisco Bay here. You've seen the effects of urbanization, the salt extraction process, and climate change combining to destroy over 90% of the original tidal marsh habitat that was found in places like the South Bay. That can be seen pretty dramatically here, where the bright green is tidal marsh in the year 1900, and then the red is the urbanized area. And you can see the dramatic increase there by 1990. So as a consequence, many of these really specialized and unique species of birds that have adapted and evolved special adaptations to cope with the pressures of the tidal marsh are all species of special conservation concern, including the federally endangered Ridgway's rail, or the black rail, which is state threatened, or these other song sparrows, Savannah sparrows, and common yellowthroats, which are all state species of special concern. And so what I've really started to want to understand is what has been the impact of urbanization, salt mining, and climate change over the past century on these specialized bird populations. And so to begin focusing on how these birds have responded, a lot of my work is focused on the Savannah sparrow. Savannah sparrows are actually one of the most widespread North American bird species. They're found from Alaskan tundra all the way down to Guatemalan highlands, primarily in open habitats. This includes Alaskan tundra, prairies and meadows back east, and of course these tidal marshes on the west coast, where you have a number of unique subspecies all the way down the coast of the Baja Peninsula and the coast of Sonora here in the Gulf of California. And this includes the belding eye Savannah sparrow, which is a state endangered subspecies found mostly in the Los Angeles and San Diego areas. In a lot of ways, this species is really well suited to addressing questions of how the birds of the San Francisco Bay Area have responded to this past century of anthropogenic change. And if we zoom in on its California distribution, there are actually 4 subspecies. There's 2 subspecies, which are adapted primarily to freshwater environments. This includes the nevadensis subspecies from Eastern California and Brooks eye from North Coast California, where they don't really occur in tidal marshes. And then you have two subspecies along the coast, which are primarily in salt marshes. This includes that state endangered belding eye subspecies. And then this, the Bryant's Savannah sparrow, the Loudonous subspecies, which is the one we find in tidal marshes here in the San Francisco Bay, but also in some upland grassland habitats like you see out at Point Reyes. And across these different tidal marsh populations, you actually see the different tidal marsh populations of the Savannah sparrow have actually experienced dramatically different impacts from humans. So you have things like in the Bay Area, in the Los Angeles metropolitan area, where over 90% of the habitat has been lost. And the same situation has occurred in places like Humboldt Bay, but there the Savannah sparrows have transitioned primarily to cattle pastures. And then you have places like Morro Bay down here just north of Santa Barbara, where the tidal marsh in that estuary is actually increasing through time. So this sets up a really nice way to compare both through space and time how these different birds are responding to a history of human activity. And then one final thing that makes this species great to study is that their historic collection of specimens are quite high. And the reason I think this dense spatial and temporal, the existence of this dense sampling spatially and temporally of museum specimens is important for addressing these questions is that museums preserve a record of population change through time. So we can look back to the 50s, the 1880s, the early 1900s or beyond, using museum specimens and understand how these animals have responded morphologically, in their molt phenology, in their genetics, or even in the deposition of soot with changing air pollution laws and how that's impacted the bird populations. So it provides this really great source of baseline data that we can compare to samples we can get from modern populations and then understand how they've changed through time in response to human activities. So to begin, one of the really easy things to measure on bird specimens is bill size. And as I mentioned earlier, one of the common adaptations you see to tidal marshes are that these sparrows have larger bills, as seen here. And this is true not just for Savannah sparrow, but a whole wide range of salt marsh sparrow species show much larger bills than their freshwater adapted relatives. And one of the reasons why we think these birds may have larger bills is that it helps with thermoregulation. And so this is a song sparrow subspecies from the East Coast that also occupies salt marshes. And here it's in a special chamber where you can crank up, slowly increase the temperature. And there's a special infrared camera that allows you to see where the bird is shedding heat. And so as you see, as it gets warmer up to above 30 degrees C, you see that the bill starts to get this bright, bright, bright red. And once you get above about here, they take the bird out and release it, and it's fine. But this is showing that these birds are using their bills to shed heat to the environment. And this is thought to help them deal with high temperatures, but avoid losing water to the environment. So normally most birds maintain their body temperatures by panting or gullard fluttering, which you see in like a lot of cormorants. And that's running air across the respiratory surfaces to cool down using water, which when water evaporates, it cools the surface. But it also comes at the cost of losing a lot of water. Another thing that salt marsh sparrow species have been shown is that they have these increased nasal conchae, which is also involved in recapturing water as it exits the bird's body. So with a greater surface area, more of that water vapor with every breath the sparrow takes is recaptured and not lost to the environment. So I was with my postdoctoral advisor, Rory Bowie, I was interested in studying how, through space and time, Savannah sparrow bills have evolved in response to different salinity and other abiotic environments, such as temperature and humidity. So across California, you see these eastern birds tend to be in hotter and drier and less, obviously, freshwater populations. And on the other extreme, you have things like the Belding eye subspecies, which is in a moderate temperature but high salinity environment. And so I took measures from nearly 950 specimens. And for every bird, I measured bill length, bill depth, and you can't really see it here, but bill width. So if you look at the top of the bill width. And I estimated bill surface area as this sort of modified equation for the surface area of a cone to approximate the shape of a bird's bill. And bill surface area basically is giving you an estimate of the total surface that bird has to radiate excess heat and stay cool. And so the first question I wanted to address is how have these bills changed through time in these 4 different subspecies of Savannah sparrow? And really the only one you need to focus on here is Alaudinus, the Bryant's subspecies, which is found in the Bay Area. And this is the only one you actually see a temporal increase in bill size, albeit fairly subtle through time. So I wanted to look more closely in this subspecies. So this plot shows variation in bill surface area across different latitudes, with historic specimens in red and modern birds in green. And what this shows is that you've actually had unequal change through time in different populations. So further north in places like Humboldt Bay, you actually see a slight decrease in bill surface area, whereas as you move further south to places like Morro Bay, you actually see increasing bill surface area through time. So this presents a nice opportunity to start looking at, well, what are the differences among these different marshes that have led to these sort of non-uniform changes through time. So I looked at a number of different habitat and like human population density and other metrics to see what might be driving this. And the best explainer was aspects of the abiotic environment. So specifically temperature, vapor pressure deficit, which is related to humidity, and precipitation and salinity. And so what this plot here shows is that populations like Humboldt Bay, which have over time gotten wetter, and these marshes have lower salinity, bill size has not changed as much as it has in places like San Pablo Bay here, where it's gotten hotter and drier, and this is a higher salinity marsh over time. I also wanted to see what effect this would have on overall water savings in the bird. So the hypothesis is that the increased bill surface area allows these birds to reduce evaporative water loss and overall conserve water. So from my dissertation, I had collected evaporative water loss data on these sparrows. And just very briefly, we would net sparrows. This is a temperature cabinet where you would place the birds and allow them to stay in their thermal neutral zone, or where basically the temperature where they're the most comfortable. And then you could measure the amount of water vapor coming out of the animal chamber as a metric of how much water they're losing. And this showed that marsh birds in generally have lower evaporative water loss than freshwater-adapted birds. But it's also importantly, this metric of total evaporative water loss, where a higher number means you're losing more water than a smaller number, is negatively correlated with bill surface area. So these larger billed birds are losing less water than smaller billed birds. So that fits the hypothesis. And if you just do some back of the napkin calculations of what the historical bill surface mean is versus the present day mean, this estimate shows that there's been a 16% reduction in the amount of water loss per day. So that would be a pretty significant reduction for a pretty small bird. All right, so these data suggest that these birds do have the capacity to adapt to a changing climate, at least in the past century. And my hope is that they could continue to adapt as things get warmer through time. However, another aspect of population response that I want to understand about these tidal marsh birds is how they've responded to the loss of over 90% of their habitat. And so this can have really severe consequences on bird populations, as has been well documented. And this is also true of the population genetics of these species. So one species that's been really long studied as to understand the population genetics of population declines in response to habitat loss is the Florida scrub jay. And then this long-term data set from the Florida scrub jay has shown that over time, you get decreases in overall heterozygosity as the population becomes more fragmented due to habitat loss, and overall increase in inbreeding. So these are really common hypothesized responses to population declines and habitat loss. You're losing a lot of genetic variation that may allow these birds to adapt to future climate change and avoid inbreeding, the negative effects of inbreeding. Of course, one way you can mitigate the costs of increased inbreeding in declining populations is through gene flow. And this has been shown most compellingly in sort of examples of facilitated gene flow, such as in Illinois prairie chickens, where introduction of birds from, I think, Kansas resulted in gene flow with the local population and increases in overall numbers of birds and also the number of eggs they were producing. And this has been shown in a wide range of bird species and guppies, bighorn sheep. And in each case, with the onset gene flow, you get an increase in population genetic size. So this would be a really great way to respond to inbreeding depression. However, on the flip side of this, fragmented and declining populations also could become more susceptible to hybridization or gene flow with a maladapted population. And this could lead to what we call outbreeding depression. So example of this is in cutthroat trout in Montana, where actually hybridization with a non-native rainbow trout led to this decrease in fitness, or the number of offspring each female was producing as the amount of rainbow trout genes were increasing in the population. So this is a negative effect of gene flow as opposed to the positive ones I mentioned earlier. And this has also been shown across deeper evolutionary timescales, like in the salt marsh Savannah sparrows. You see that in a trait that's associated with adaptation, with increasing gene flow from freshwater adapted populations into the salt marsh adapted population, you get the decreasing divergence in this trait. To me, it means it's really important to try to understand the different dynamics between selection and gene flow to understand how these populations may respond to habitat loss and may recover from habitat loss as well. So with these same Savannah sparrow specimens, I was interested in looking at population genomic responses. And this is a really great species for doing that because you have both these freshwater adapted populations and these tidal marsh populations. So you have both the opportunity for gene flow from populations outside the Bay Area, but it could also have negative impacts given that these are adapted to different salinities. And so I really wanted to address three questions with this, with these Savannah sparrows. One is, how has genetic diversity changed through time? how has habitat loss impacted population structure and gene flow over time? And importantly, what impact has this had on genes that may be associated with adaptation to tidal marsh habitats? And so to analyze these data, I used what's called an exon, or to generate this data, sorry, I used what's called this exon target capture approach. And without getting into too many of the details, exons are the regions of the genome that code for the proteins that do all the work in our bodies. And exon target capture is basically custom DNA probes that will target only those regions in the genome, and often in small fragments. And this is what's really ideal about this approach for historical museum specimens, is it allows you, because these over time, as DNA degrades and becomes smaller and smaller fragments, but these little custom probes can still allow you to sequence a good chunk of the genome. And so I developed a capture approach using the white-throated sparrow genome as a reference. It's one of the best genomes out there for any sparrow species. And I ended up with a number of target regions or exons from over 14,000, from nearly 14,000 genes, and a number of regions that are non-coding as well. And then to get DNA from specimens, what I did is cut a little bit of the dried toe pad from the specimen. And from there, I did a lot of lab work, and then sequenced it on one of these Illumina NovaSeq, machines which generated millions of these reads, no, billions of these reads actually, which you can then assemble to a reference genome and understand, give you the genetic data from all specimens from, collected from about 1890 to the present. And so to address that first question of how genetic diversity has changed through time, I first want to look at how it varies in space. So this plot just shows spatial patterns of genetic diversity, which have been pretty similar through time. And so these darker brown colors tend to show lower regions of genetic diversity in California, whereas these darker blue regions show higher genetic diversity. So in general, you have these southern populations, like this is Orange County, Los Angeles, having lower genetic diversity than Northern California populations. So next, by comparing the historical specimens to the modern specimens, I could tell how much these two common metrics of genetic diversity have changed through time in these different populations. What you see is, again, in these southern populations in Newport Bay or Orange County and Morro Bay, you have a significant decrease in genetic diversity through time. Whereas here in the Bay Area, you actually don't see any change in genetic diversity over the past 120 years or so. And this is kind of surprising because, as you might remember, both Newport Bay and the Bay Area have experienced similar levels of habitat loss. And so what might be explaining this? So to explore this further, I looked at this, used this program DieStruct, which is a way of looking at population structure using some sophisticated models. And importantly, DieStruct, as opposed to other similar methods, allows you to account for when the sample was taken. So the historic samples are then the ancestors to the modern samples. What this shows is that historically in California, you had populations of Savannah Sparrow that were showing distinct genetic structure in Southern California. In the Bay Area, showed another population genetic cluster. And then in Eastern California, had a third population cluster. And then some of these populations, like this is Northwest California, shows these two different colors, which suggests it's has ancestry from both Eastern California and the Bay Area, and similar to Morro Bay, which also shows relatedness to both the Southern California population and the Bay Area. In the modern data set, birds mostly where genetic samples were collected after 2000, you see similar patterns, Southern California, Bay Area, and Eastern California. However, if you look at the San Francisco Bay Area, you see that these populations here have a much greater proportion of ancestry from Eastern California. And so what this suggests is that there has been, over time, an increasing level of gene flow coming from Northern and Eastern California into these Bay Area populations. So what this suggests is that the stable genetic diversity through time in these Bay Area birds may be due to the fact that you have this influx of immigrants from outside the Bay Area that helps maintain the population genetic diversity over time. However, as I mentioned earlier, there may be negative consequences to this gene flow. And so to explore this further, I want to understand what the loci under selection were, or the genes under selection that may be associated with adaptation to tidal marshes were, and what the consequences of gene flow may be on this. So to do this, I used a common metric of genetic divergence called FST, which is just when you have two populations, population A and population B. FST is a measure of how divergent these populations are genetically. And you can split the genome into a bunch of tiny regions and estimate FST for each of those regions. And what you get is these, and when you compare the FST across the genome, you get these uneven surfaces where some parts of the genome have very high FST and some regions have FST approaching 0. And this dashed line is a somewhat arbitrary threshold that tells you FST, regions of the genome that show FST above this threshold are likely genes that are under selection and associated with adaptation to a specific habitat. So I did this for, I calculated FST for between historic populations in Eastern California, which are freshwater adapted, and these Bay Area populations, as well as these other tidal marshes. And I used threshold to identify regions of the genome that may be under selection associated with adaptation to tidal marshes. I then did the same thing with the modern samples and just subtracted FST from the historic FST from modern FST to get a sense in how FST has changed in these regions that may be under selection. And so what the results of that can be seen here. Where in blue you see parts of the genome that aren't considered outliers. They're probably not involved in adaptation to tidal marshes. And you really see no change through time. Average change in FST is 0. However, in orange, you see for each of these tidal marsh populations, on average, you have a decline in FST, suggesting it's becoming more similar to, these populations at these regions are becoming more similar to the Eastern California populations, where I've hypothesized there's influx of immigrants. And so to look at this a little more closely, I want to just focus in on one gene, and that's this estrogen receptor 1. This was it was the top outlier in both these Beldingi Savannah Sparrows historically and in the Bay Area Savannah Sparrows. So this is a strong candidate for a gene that is under selection and important for adaptation in tidal marsh environments. And importantly, it has known, estrogen receptors are involved in a huge number of different functions in the body, but importantly, it has a number of key osmoregulatory functions, including these estrogen receptors regulate gene expression in the aquaporin gene. And aquaporins are really one of the most critical proteins involved in concentrating salts in the urine in the collecting ducts of birds. So this is a strong candidate for influence on osmoregulation birds. And what we see is that over time in the Bay Area, this gene has decreased in FST by 0.16, and FST is on a scale from 0 to 1. So it's a pretty significant decline in FST, suggesting that this influx of immigrants from Eastern California has had negative consequences on adaptation in these salt marsh birds. So with that, I'd like to wrap up the talk and say that although immigration may contribute to stable levels of genetic diversity over the past 125 years or so in the Bay Area, there is some evidence that gene flow from freshwater adapted populations may be a concern for these salt marsh adapted sparrows. However, I also showed with the bill traits, the bill surface area, that these tidal marsh specialist bird species do appear to have some capacity to respond to future climate change. And in some, I think this really highlights how valuable museum collections are in providing abundant opportunities to analyze replicated time series of specimens, which can really provide important insights into how populations are responding to anthropogenic change. Not to intend on a high note, I think while I show that there is potentially this negative consequence of increasing immigration from outside the Bay Area, you do have lots of really great salt marsh restoration projects going on all over the Bay Area. And I really encourage you to go out to places like the Hayward Regional Shoreline, the Palo Alto Baylands, some of the salt marsh areas around El Viso, and explore these habitats. And also, hopefully, as we restore more and more marsh, which is a major focus of a lot of different conservation groups in the Bay Area, that hopefully that will help reverse these trends. And so with that, I'd like to acknowledge some of my collaborators, especially Rory Bowie and Jen Walsh, Lydia Smith, who helped with all the lab work and field work. A lot of this project was funded by a postdoc grant from NSF. And then all these natural history collections were important for both aspects of my project. And we wouldn't have insights into how these birds have changed through time without the hard work of curating these collections by the staff at these institutions. And so with that, I'd like to thank you for coming and open it up for questions.
Awesome, thank you so much, Fred. That was really interesting. And I know that every time I go out and see Savannah Sparrows in the marshes, I'm going to be thinking about all this great work that you did with them. So thank you. So the first couple of questions were related to other species. So we can first do that. So we had someone ask about whether there are also changes in the common yellowthroats that inhabit tidal marshes.
I would love to address that question, but yeah, I don't know yet. I am working a little bit on, we're starting to work more on the song sparrows that we have here in the marshes, and hopefully many of you saw Chloe Mickles talk earlier in this season. But yeah, yellowthroats are another species I would love to look at and see how they're changing through time. But it sounds like SFBBO has some blood samples from these yellowthroats, so maybe we can work on that in the future. It could be really great, interesting to see if similar patterns emerge between the species or something completely different.
Nice, yeah, so stay tuned. Maybe we'll have an answer soon or in the future sometime. And then we had someone who's joining from Florida, or from Louisiana actually, who asked if you're familiar with seaside sparrows and if you know anything about them or can comment on them.
Yeah, so seaside sparrows are great. Yeah, I spent 4 years in Louisiana as an undergrad, so I love seaside sparrows in Louisiana tidal marshes. Yeah, seaside sparrows are one of the most adapted sparrows to salt marshes. So we've done some estimations of colonization time based on divergence, and they're by far the deepest divergence. We think they've been in salt marshes longer than any other species. They do show a lot of the sort of common physiological and behavioral responses to salt marshes, at least in what's been studied. Yeah, it's not a species I've worked on. Well, one paper. That was the one where we described how deep the divergence is. But yeah, and I know some people at LSU are planning on working more on them, but I think in part looking at the sort of toxicology in response to things like oil spills in the Louisiana tidal marshes. So, but yeah, fantastic species.
Nice, thanks. And then related to how you were talking about the bigger bills, question was, do the birds also tend to have bigger legs and lose heat radiatively through the legs too, in addition to the bills?
Yeah, actually the Belding Savannah Sparrows do have longer legs. So yeah, that could be definitely a possibility. And yeah, it'd be great to look at that. We don't actually have any of this like infrared camera data for the Belding Savannah Sparrow, but hopefully future work. But it's always tricky. So one of the, like any of these morphological characters, they never are involved in just one thing. So it's also really critical for locomotion, and these things are running through mud, which is a completely different substrate than like a lot of sort of Savannah sparrows in the Sierras are going through. And bill size is also maybe responding to different dietary influences. Like these Belding Savannah sparrows actually eat crustaceans. So they've been known to eat fiddler crabs. They're not just eating seeds out in the salt marshes. So they eat a lot of invertebrates, including these hard-shelled crustaceans. But with some of my work would change through time, I also look for use metrics of changes in habitat, and that didn't seem, which may be correlated with diet, and that didn't seem to influence the patterns I saw. But yeah, great question.
Yeah, cool. Now I just really want to see a Savannah sparrow eating a crab.
Yeah, you can watch them at like low tide, that like with all the crabs, and they'll chase around. There's even an old paper from like well, not that old, it's from like the 80s, of a Savannah sparrow chasing a female fiddler crab around and pecking eggs out of her as she was laying them and eating them. So yeah, cool. Next time you go to Los Angeles or San Diego or something.
Mm-hmm. And then so related to genetics, does introducing individuals to increased genetic diversity also lead to populations becoming more similar to each other? And is there a downside to that? So you did address some of that in your talk. Is there anything else you want to add?
Yeah, that's definitely one of the chief concerns. And so and this seems to be a case where that's true. And maybe it's because the habitats are so different. And gene flow has this well-known phenomenon that does homogenize, but it also eliminates like a lot of the negative impacts of inbreeding depression, such as like all these recessive diseases and things like that come to the front when you have low genetic diversity. I showed briefly the guppy example. Those were in pretty dramatically different habitats, and you actually didn't see a homogenization of these outlier genes that may be associated with adaptation to these different high predator, low predator streams that the guppies were going between. But there's still not a lot of data out there. There's a lot of great examples of genetic rescue, but less of this outbreeding impact, but we do think it's important. Certainly merits lots more study.
And then another question that perhaps needs more study is, has climate change and fires contributed to the genetic changes in the San Francisco Bay subspecies?
Climate change definitely could. So climate change has a lot of different impacts on adaptation to tidal marshes, including here in the Bay Area. So one is sea level rise. That means that tides are getting higher and inundating more and more of the marsh. So that could also contribute to habitat loss, but also contribute to the marshes getting saltier and exacerbating some of these selective pressures. But since you have these freshwater inputs, it's also drought and reduced snowpack due to climate change is also reducing freshwater, well, plus all the agricultural pull-off of the, which I guess is slightly separate from climate change. So these all different factors contribute to the changing salinity patterns in the Bay Area. And there is some evidence that salinity has increased over time, though most of that's from like out in the open channel, not necessarily in the marshes themselves. And so, yeah, it would be really interesting to see what genes have changed over time within the Bay Area. Unfortunately, one thing that makes it tricky is we have genes coming from, or versions of the genes coming from elsewhere. So it's hard to tease those two things apart. Are these new variants due to immigration from other populations, or are these things that have changed within the Bay Area? So I haven't quite figured out how to tease that apart yet. So maybe in the future I will have an answer to that question, or a more direct answer to that question.
Yeah, that sounds like a challenge. So first, before we continue with questions, I do want to let everyone know it is 6 p.m. over here on the West Coast. So if you need to log off, I want to thank you for joining us. But Phred, if you have a couple more minutes, we do have a couple more questions that we can get to.
Yeah, definitely happy to stick around.
Yeah, so another question that we have is, why might the Rostratus subspecies have a larger bill than the belding eye or other coastal subspecies?
Yeah, great question. So it's more than just salinity. So there's this so since the hypothesis is that this is a that they're using these bigger bills to radiate more heat and thermally regulate belding. Belding Savannah sparrows are in cooler environments than the Rostratus subspecies, which I don't know if you've ever been to the Sonora desert in the middle of summer, or especially the Colorado River Delta where the tidal marshes are, these sparrows breed, but it's one of the most miserable places I've ever been. It's like 40 degrees Celsius. I don't know what that is, 120 degrees, 115. And these birds are breeding in there and they have no access to fresh water. So the hypothesis is that they have this really large bill to radiate excess body temperature while conserving as much water as possible. And they do, I don't have data from there, I have data from further south, but they do also show lower levels of evaporative water loss than even the belding eye or some of these birds in the Bay Area. So it does seem to be holding up so far. And if you look at the song sparrows in the Bay Area, you also have this intersection of temperature and salinity. So the place where the Bay, so there's the three parts of the Bay, the CSUN, San Pablo, and San Francisco. And so you have three subspecies of song sparrow, and they vary in bill size as well. And that variation in bill size is best explained by temperature. So it gets hottest in CSUN Bay, like over by Fairfield. And that is the largest billed of the three song sparrow subspecies. So there seems to be, they're in this dehydrating environment, but the maximum temperature is interacting with that lack of water to drive the evolution of larger bills.
Oh, very interesting. And then the last question I'm seeing for now is from Dan, who is one of our biologists here at SFBBO. He says that Savannah sparrows also nest in grasslands and Santa Cruz Mountains, which aren't salt marsh and probably not an available habitat historically. Are they maybe shifting their breeding area?
Yeah, so historically there's been, this subspecies, the stronghold has always been the Bay Area, or the tidal marshes of the Bay Area, but they're from like Monterey Bay up to Humboldt Bay, they're in these sort of coastal grasslands in the fog belt. So like Point Reyes, they're really dense there, even though it's not necessarily salt marsh. I haven't seen the ones in the Santa Cruz Mountains, so I'd like to go check those out. So one that could be a source for some of these genes from freshwater-adapted populations. I haven't explored how divergent they are genetically at different regions, or maybe they're the same. So that would be something I would like to explore further. Yeah, and if they're expanding, it's possible. Humboldt Bay is really interesting in that historically the Savannah sparrows there bred in tidal marshes, but the available tidal marshes are basically gone and they transition to cattle pasture. So it's possible that they can also just transition to a new habitat. And that could be accelerated by immigration from these freshwater adapted populations. So certainly something I would love to look into more. And I'll have to go explore those birds in the Santa Cruz Mountains.
All right, well, I think that was the last question. So thank you so much, Phred, for your talk and for giving all those great answers to such a variety of great questions. So thank you so much. And thank you again to everybody for joining us. Phred, is there anything else you want to share with everybody before we sign off?
Not really, but yeah, go out and explore tidal marshes. They're great. I mean, yeah, not just the sparrows, but all the birds that occupy these environments are one of my favorite habitats and great things about living in the Bay Area. So.